She's the chair of the United Kingdom's Food Standard Agency. Please welcome Susan Jebb.
Thank you so much for this kind invitation to speak at this really fascinating conference. I'm especially pleased to join you as it's a kind of perfect coming together for me of my own scientific research interests. And I have a research team at the University of Oxford who do quite a bit of epidemiological research ourselves, as well as some clinical trials. But it also combines that with my interests as a regulator, as chair of the UK Food Standards Agency. So as you heard earlier from Erica, the FSA is an independent, non-ministerial government department, and we're responsible for really protecting consumers' interests in relation to food. And since we left the EU, the FSA's had a greatly expanded role in the food system in the UK. So we're responsible for risk assessment, for risk management, and for risk communication. I'm really pleased that that's meant that we've been able to greatly expand the number of scientists we have in the agency. We've just about doubled the number. Not nearly as many as I'd like, of course, but nonetheless, we're on the way. But I'm really conscious that we are a regulator with a budget of about £130 million a year. But we're responsible for keeping food safe in a food industry that's worth £240 billion a year. And that difference tells me that we can't keep food safe on our own. I well know from my own scientific research that science is a global enterprise and that we really have to learn from the best in the field. And that's why conferences like this are incredibly valuable to share those experiences. We in the UK FSA really value our international links, and we're here at this meeting to learn from you. And I hope also to share some of our expertise to solve some of the problems which are being faced internationally. Food's a global business, it's a global trade, and we're not going to be safe until all of our food systems are safe. So epidemiological research is an absolutely vital part of our risk assessment. You don't need me to stand here and tell you that. And so in the FSA, we're very interested in using the latest epidemiological methods. We're interested in how the principles of epidemiology get incorporated into our risk assessment assumptions. And as chair of the FSA, I'm keen to ensure that we and other regulators have access to the best evidence to carry out that risk assessment. And as you heard in the example earlier, that sometimes means us looking overseas at work that's been done elsewhere, because the risk assessment's generally going to be the same. Risk management may sometimes differ, but risk assessment is generally a pretty common business. One of the things we've been really trying to do within the FSA is to draw together all the different scientific disciplines so that we don't have everybody working in silos if they're worried about microbiology or they're worried about radiological contamination. And we've been doing some work with our independent science committees recently to try to develop some guidance on how we can best synthesize and integrate different types of evidence in some of our risk assessments, particularly in relation to the incorporating toxicological information into chemical risk assessments. So what are some of the sort of newer things that I think are interesting? First, I want to reinforce the point that Erica made about the importance of whole genome sequencing. I think this is being increasingly used in our incidence response work to identify and differentiate between different bacterial and viral strains, and also to explore how foodborne pathogens and antimicrobial resistant microbes move between environments, farms, and foods. I've no doubt this is going to be increasingly important. You heard the example that Erica gave about the use of whole genome sequencing in really trying to get to the bottom of the listeria outbreaks that we've had recently. But there was another one which you might be familiar with, which was the outbreak of salmonella in Kinder eggs. So this was an outbreak which affected, there were cases across Europe in the run-up to Easter last year. And through whole genome sequencing, we were able to link cases and to directly trace that back to Kinder eggs and other chocolate products that were being produced at the Ferrero factory in Belgium. That prompted a product recall not only in the UK, but in 98 other countries. And coming as it did, just before Easter, I think it undoubtedly prevented many of our children from becoming sick. Erica also mentioned our PathSafe programme, Pathogen Surveillance in Agriculture, Food and the Environment. Again, that's using DNA sequencing technology to improve the tracking, the detection and the tracking of foodborne disease and antimicrobial resistance. The thing that I like about PathSafe is it's a really collaborative, interdisciplinary project. It brings together policymakers with people in industry and also with a number of different academic groups. There are over 50 collaborators working on more than 30 projects, very much in a One Health approach. And there's a PathSafe website if you want to follow the work that project's doing. I'm really excited about some of the work we're doing at the FSA. It's been great to hear some of the other new ideas which have come up at this meeting. I have to say, I share your pain about risk of bias assessments. If only there was an easy way of doing that. But there is so much valuable work going on, and we've got a lot to learn from each other. I hope that this meeting has been an opportunity, as conferences are, to renew links with old friends and colleagues, and I hope also to make some new connections. I really want to thank EFSA and BFR for bringing us all together today and for giving me a fantastic opportunity to come and visit Berlin. I did miss the workshops this morning because I had some great meetings both with BFR and more recently at BVL. There's quite a lot of traffic in Berlin, so it took me a little bit longer to get back than I hoped. In the UK, we're really keen to expand our international collaborations. And so if you are interested in setting up reciprocal visits to come and see more of what we're doing, or if you want to think about joint projects, we'd really welcome a conversation. So please do come and find me or one of my FSA colleagues sitting over here afterwards, or just send us a message later. I'd be really keen to work with you. Thank you very much.
Thank you so much, Susan Jebb, for this short statement of yours. Any very, very urgent question from online or in the hall? No? No worries, all good. Thank you. Thank you so much also for the invitation to stay in contact. And let me pass the baton directly to our next speaker. He is chair of the EFSA panel on plant protection products and their residues and professor of toxicology at the University of Granada. Please join me in welcoming Antonio Hernández Jerez. Welcome.
Well, thank you very much, EFSA and BFR, for inviting me just to make a brief statement, which is basically the same that you can find in the program of this conference. So, epidemiological studies are valuable for risk assessment, but if there is a couple of things are fulfilled, these epidemiological studies have to undergo previous risk of bias assessment. And also, the second is that they are not a standalone stream of evidence. They should be integrated with other lines of evidence in order to provide, let's say, a more general conclusion about the available information or available evidence that is assessed. So, human epidemiological studies, observational studies should be assessed for reliability, but also for relevance. And in this case, might be used for hazard identification and to a lower extent for hazard characterization. Note that in Tomorrow, there will be some speakers that will address what are the different tools to perform these critical appraisal of these studies. In the case of EFSA, we have used particularly the NTP OH80. So this is the National Toxicology Program Office of Health Assessment and Translation. This is the one in which we have gained more experience, but for sure there are other tools that can be also be used with similar results. The problem of the risk of bias, internal validity, or systematic error is that it can lead to deviation of the truth. Deviation of the truth in the results or in the inference. So eventually leading to overestimation of the risk or underestimation of the risk. So for this reason, this is a key important step previous to the consideration of observational studies in risk assessment. The other appraisal important in our view, and we are using in the EFSA, is the assessment of relevance, the relevance of these studies. Relevance is inversely related to the risk of bias. So higher risk of bias means less relevance of the study. But the relevance is also related to the external validity, to what extent the results are extrapolable to the general population where this human sample has been drawn. And also very important relevance is what is the impact of the findings of that study on the current risk assessment. Produce an important change or not? So at the end, the epidemiological studies, as has been mentioned earlier, everybody knows, try to make an association between exposure and an effect, a human health effect. However, what occurs in between is most of the time unknown. So we need to provide something that gives more biological plausibility, in other words, gives higher possibility of causality, causal association. How can we give this biological plausibility? Using other streams of evidence, using also in vivo studies and also in vitro studies of NAMs. If we use all this line of evidence, human, in vivo, and in vitro, in an integrated way, using a weight of evidence approach, using the adverse outcome pathway, for sure that we can improve the risk assessment. And this is, and EFSA, this is my last comment, EFSA produced a scientific opinion, the PPR panel in 2017, regarding how to integrate epidemiological studies into risk assessment and how to integrate with all lines of evidence. And this has been expanded by the scientific committee, and Thor will maybe mention later on. It has been expanded to other EFSA sectors by developing updated guidance in this regard. That's my briefest statement. I hope you have stick on the 5 minutes.
The briefest statement. Thank you very, very much. Urgent questions, one or two? No? No. So thanks for sharing your view and opinion on the topic with us. And that brings us to the third speaker for this session. Thorhallur Halldorsson, I hope I pronounce it correctly, is professor at the Faculty of Food Science and Nutrition at the University of Iceland, and he is member of the EFSA's scientific committee, where he's the chair of the working group on EPIC studies. So, very much welcome. The lectern is yours.
Thank you very much. I'm a bit surprised that I'm asked to give a brief statement because people who have been in meetings with me know that I can talk very rapidly and a lot, but I'm not particularly good at giving statements. But I want to talk about the use of epidemiological studies in risk assessment, and I think the discussion here today about biases, about exposure and other things, and at the end of the day, I think it boils down more into learning from each other to some extent. There's been a lot of talk about how we can do, in the old days when I was starting, how can we make epi studies better? We need better studies. They have to be different. But at the end of the day, studies are done as they are done. And I think, for example, epi studies are not done to be used in risk assessment all the time. You do occupational studies on pesticide exposure to prevent workers or from public health issues, not because you want to examine whether the active ingredient in a certain plant protection product is causally related to some disease, which we should not talk about today. Anyway, and I think, if you understand that, you understand the type of studies that you're getting sometimes. And can they be used? Yeah, of course, because like you heard with the mobile phones, yeah, you have all the signals. Maybe you didn't have to do all this bias assessment. The signals were already there. Don't read too much into a single study. Use the totality of the evidence, and maybe the answer was already there. Maybe. And it's about learning from each other. And I remember I come from nutrition, and I was working with a lot of animal people on nutrition. And I had the same doubts. Why are they feeding animals something that they shouldn't be eating overall? And why are they using bonus doses of fish oil? So, I mean, we have all these doubts about our disciplines, but working with people, you learn to figure out, yes, this is the time to use the animal study. And this is how we design studies together. And I think we are getting there slowly in risk assessment. I remember I didn't understand EFSA opinions. Why are they writing about epidemiology in that way? At least if you want to convince the public, write a little bit differently. Don't come up with a list of 50 limitations. Nobody buys that. And we also learn from each other within medicine and sort of, I remember from nutrition, the EU stopped funding almost observational studies. Now you should play with the big boys and do sort of randomized controlled trials. That's fine. Some people ended up randomizing thousands of people to one antioxidant, and people got cancer because the observational evidence was there. If you eat vegetables, get less cancer, but a single antioxidant bolus dose in a pill for a few years, I mean, maybe it's causally related to small increase in cancer, but is this what we are talking about? Is this public health, or is it an academic exercise? And there are plenty of, and the other thing, I've been working for 5 years now on the Nordic Nutrition Recommendation. I was in the previous one as well. It was all about RCTs. Now everybody stopped talking about the RCTs to the same degree. They are extremely useful. But does it help to randomize people to different lifestyle and see whether they die less prematurely than the other group, and you have no compliance in the end, and there is no conclusion? I think in terms of our public health advice for macronutrients, we are doing a pretty good job just relying on observational studies. And this is also about relying. We need all these types of studies, but we need to understand how to use them, when to use them, and when not to use them. And that brings me to the final, 2 points on the risk of bias. It's extremely useful, it is helpful, but it doesn't solve anything because the lowest risk of bias study, the best conducted one, can be absolutely biased because we are assessing risk. And high risk of bias study can actually be very useful. So it's a checklist, and we need to do it. It absorbs maybe too much of our time, but it's helpful. Same as study protocols and everything is helpful. It makes us think before we do. But it's only one of several things that we look for. And we need to use the totality of the evidence rather than being picky. And maybe being picky and picking the best study comes from the experimental field, where maybe that's the most logical thing. And so we need to use that, and in the future, I also think we need to be very careful when we use epidemiological studies for risk characterization. The same procedures do not apply. It's better to use an integrated result from several studies, and there are numerous examples where we actually overdo it. And if I can talk about the exposure a little bit, because that was a lot about that. It's nice to have a framework for exposure. Rapidly eliminated chemical can be meaningless, but if you have another prior, like where people are working, how they behave, then the spot urine makes perfect sense. And I think we should be careful what we wish for, because I've once worked with 24-hour urine, and I've seen how some studies with repeated 24-hour urines are interpreted and over-interpreted as being an example of good exposure. Now, when you ask people to sample 24-hour urine, you get a brown tank, and you're supposed to discharge everything over 24 hours into that tank. You don't go to a club with that. You don't go to a restaurant with that. You stay at home and you eat differently. So it's like the Heisenberg uncertainty principle. You bias your estimate by simply asking people to do too much. And so we need to be careful and we need to be flexible, and that's epidemiology. Thank you.